Hello, my name is Johan Hornbacher and I'm a PhD student at the Institute of Botany at the Leibniz University Hanover, and this is the video for my poster, Indolic glucosinolates are a source of auxin in drought-stressed Arabidopsis thaliana plants. So usually glucosinolates are degraded upon herbivoric attack by thioglucosidases, for example TGG1, TGG2 and PAN2. And because of the repellent nature of their breakdown products against biotic stressors, changes in glucosinolate contents are obvious and were reported many times. But contents of glucosinolates also change due to abiotic factors. For example, it was shown that the contents of aliphatic glucosinolates were changed in drought-stressed plants and that they were involved in stomatal closure and therefore water homeostasis. Indolic glucosinolates, on the other hand, can be degraded into the auxin indole-3 acetic acid. If nitrile specifier proteins, NSP1 and NSP5, are present upon the degradation of indolic glucosinolates with myrosinases, indole-3 acetonitrile is formed. Indole-3 acetonitrile can be degraded to indole-3 acetic acid in the presence of nitrilases, nitrulase 1 and nitrulase 5. Higher levels of the auxin indole-3 acetic acid were shown to be beneficial when applied to drought-stressed plants because of the regulation of root growth. For the experimental setup, I used 5-week-old Arabidopsis thaliana plants that were grown on soil, and I exposed them to 5 days of drought stress by holding the water salt content at 40%. Health indicating parameters like leaf water content, as well as the expression of drought stress indicating genes like P5CS1 were analyzed. Additionally, expression of genes involved in the synthesis, hydrolysis, and rearrangement of glucosinolate hydrolysis products were investigated. And of course, contents of single glucosinolates in leaf samples were analyzed. Our analyses were based on 3 biological replicates consisting of 3 plants each. So I observed that the leaf water content in drought-stressed plants is much lower compared to the control plants, and that the expression of P5CS1, the drought stress indicating gene, is much higher in drought-stressed plants compared to the control plants. Glucobrasicin contents, as well as indole-3 acetic acid contents, are much higher in drought-stressed plants compared to control plants. And finally, the expression of NSP1 and NSP5 are higher in drought stress compared to control plants. So the lower leaf water content, as well as the higher expression of P5CS1, clearly indicate that the plants were successfully drought stressed. In contrast to other glucosinolates, the higher contents of glucobrasicin indicate their special role in drought-stressed plants. Simultaneous higher expression of NSP1 and NSP5 indicate that the reaction of the breakdown of glucobrasicin is shifted towards nitriles, yielding indole-3 acetic acid at the end. Also, higher contents of indole-3 acetic acid in drought-stressed plants indicate the importance of this growth-regulating hormone in drought-stressed plants. So, in times of drought, plants could be at an advantage if they use the pool of indolic glucosinolates to yield indole-3 acetic acid instead of synthesizing auxin from tryptophan directly from the other pathways. Thank you for your attention.